Hi, and welcome to this guide to the Depth in Biology June 2017 paper. The goal of this guide is to help you understand the questions, see which theory to apply, and gain full credit to all of the questions. Let's get on with the paper. So question 1 shows you two drawings of plant cells. Now each cell is observed using a different type of microscope. So the cells are not drawn to scale, and the question asks you to give one piece of evidence from figure 1.1 that supports the claim that cell B was observed using an electron microscope. Now if we look at the picture of cell B, what can we see in there that will tell us that it was observed with an electron microscope? And what we can see is details within the organelles. So you can see thylakoid membranes, you can see ribosomes on the rough endoplasmic reticulum, you can see nuclear pores. Now these are all things that you need to have a very high resolution in order to see, so they're only possible to observe them with an electron microscope. So the evidence that you can quote is that you are able to see the details within the particular organelles. And the mark scheme supports this. So they give you a mark if you said that the resolution was higher or the resolution was better. Therefore you can now see grana in the chloroplasts, thylakoids in the chloroplasts, cristae in the mitochondria, the rough endoplasmic reticulum, you can see ribosomes. All of these things are evidence from the figure that would support the fact that it had been drawn using an electron microscope. Now the question goes on to ask, give one way that an image produced by a laser scanning confocal microscope would differ from that produced by an electron microscope. Now you've got to be careful answering this question because they are asking about the difference in the techniques of laser scanning confocal and electron microscopy, but they don't want you to talk about the detail of how the microscopes work. They want you to talk about the resultant image. So the resultant image from a laser scanning confocal microscope will have a lower resolution. It will be able to see movement because you can use living specimens, and also you can look at different depths into the sample. So important points here: it's got a lower resolution than the electron microscope. You can fluorescently tag particular molecules and then you detect the fluorescence emitted when you shine a laser on it. You can see movement, which you can't see with an electron microscope because the specimens must be dead. And you can see different layers and different depths of the sample. So remember, the question is asking about the nature of the image, and it also needs to be a comparative statement because it's saying give one way that the image differs in a laser scanning confocal microscope to that of an electron microscope. So meiosis is a type of nuclear division, and we show a photomicrograph of a plant cell undergoing meiosis. And they ask you to identify the stage of meiosis 1 shown in the cell labelled A. And here is cell A. So what can we see? We can see condensed chromosomes starting to appear, but we can also see a nuclear membrane that appears to be still present. So this means that interphase has occurred where the DNA has replicated and the chromosomes have started to condense and appear more visible, yet you've still got the nuclear membrane present, so you can conclude that you're dealing here with prophase. So based only on your observations of figure 1.2, state two reasons for your answer to B part 1. So you have to discuss something that you can see within that cell that tells you that it's prophase. So the answers that you're looking for here hopefully are that you can see condensed or visible chromosomes, yet they are not on the equator, they are not organised. You can see a nuclear envelope. You can see that the chromosomes are separated from the cytoplasm and there's no visible nucleolus. Now, another stage of meiosis is metaphase 1. So in metaphase 1, the homologous chromosomes line up as bivalents on the equator, and they line up with independent assortment. That means you don't have all of the maternal chromosomes on one side and all the paternal on the other. It's a random order. They line up in pairs, so chromosome 3 with chromosome 3, chromosome 4 with chromosome 4, but they line up in no particular order. And that gives us genetic variation. So let's have a quick look at how that can happen. So if we imagine we had a 2N cell, let's go over here and let's try and explain it. So you've got N meets N, so sperm meets egg, and each of those will have, say, 2 chromosomes in them. And then we produce a cell which now has 4 chromosomes in it through fertilization. So this is a 2N cell, 2N equals 4, N equals 2. There are 2 chromosomes from each of the haploid cells. So this is now a diploid cell. Now at some point this needs to divide by meiosis. So within that cell there are 1, 2, 3, 4 chromosomes. Now interphase occurs and then prophase, so we end up with a cell with the replicated chromosomes, which look like this. So a chromosome 1, another chromosome 1 from the other parent, a chromosome 2, and the other chromosome 2 from the other parent. Now, if we consider that they might have alleles of a gene, so the A and so the big B and the little b, well, then that would have replicated so that 2 chromatids would be fairly similar. We'll ignore crossing over at the moment, and so we would have BB and little b, little b. Now when they line up on the equator, you've got the homologous pairs lining up as bivalents. So when they get pulled apart in anaphase, you end up with one of each of the chromosomes going that way, and one going that way and that way. So that the cell that goes then into meiosis 2 will be this one or this one. So you'll have a chromosome 1 and a chromosome 2, and you'll have a copy of the A gene and a copy of the B gene. But what's important to remember is that they don't line up in any particular order, so that these could be the other way around and these could be the other way around. So that the alternate solution is that we could have this, but we could have the B chromosome 2 the other way round. So now this is what goes into one cell, which goes into meiosis 2, and this goes into the other. So then this would give us this combination of alleles, and this and this and this. So what we're doing here is independently assorting the chromosomes, and this leads to genetic variation in the gametes. Now, explain means go into quite a lot of detail and refer specifically to how the homologous chromosomes are organised and how that increases genetic variation. So what is the consequence of the independent assortment of the homologous chromosomes? So the mark scheme is looking for you to say specifically independent or random assortment of the homologous chromosomes, which is the name for the arrangement, and then to say what that is, that they line up on the centre of the cell on the equator, and that they line up with maternal or paternal chromosomes and one in each pole, so that one facing either pole, so one goes into each of the 2 daughter cells. So into your sperm or your eggs, you either put your mother's chromosome 1 or your father's chromosome 1. And then Each of the chromosomes of a homologous pair can be genetically different or contain different alleles. Now, the use of stem cells is being evaluated for the treatment of certain human diseases. Name 2 potential sources of stem cells, or human stem cells, and for one of those sources, describe an ethical issue associated with the use of stem cells. Now, you've got to be careful with this answer. It's a 4-mark question. It's very easy to get 2. It's quite hard to get 4. Now, they're asking you to name 2 potential sources. So this is of unipotent, multipotent, pluripotent stem cells. And then for one of those sources, and it has to specifically refer to that source that you talked about, discuss an ethical issue associated with the use of stem cells from that source. So you do have to take care with the phrasing of your answer here. Now, the sources that you could be thinking of would be things like umbilical cords, very early fetal cells, bone marrow. Let's have a look at the mark scheme. So 2 marks for the sources, which would be embryonic cells, fetal cells, umbilical cord, bone marrow, or induced pluripotent stem cells from taking a somatic cell. In fact, it would be a unipotent stem cell and turning it into a pluripotent stem cell. And then the ethical issue has to relate to that particular source. So if you talked about embryonic stem cells, you might be thinking, well, in order to obtain those stem cells, you have to destroy the embryo. And that leads to a debate about when life begins and the fact that the embryo can't consent to its own destruction. You could go on to talk about the fetal tissue obtained from miscarried or aborted fetuses, which is controversial for some people. And then you could talk about the harvesting of bone marrow, which they say is painful and risky. I don't think it is anymore, but I could be corrected there. And the babies being conceived specifically to give a bone marrow match for a sibling, and that you should then show that you've made an opinion based on science of this ethical issue. And that's what you had to do to get 4 marks. It's a bit of a challenge, and remember that you had to make sure that the ethical issue was linked to the source of the stem cells that you had mentioned in the first part of the question. A group of students wanted to observe the position of the xylem vessels in the leaf stalks of celery. Now, it asks you to describe a procedure that they could use to do this. Now, describe means what would you actually do in order to see this. So hopefully you might think, well, I'll take the celery stalks and I'll put them into a coloured liquid, and the coloured liquid would rise up through the celery stalks, and I then cut a thin cross section through the celery, and I would then be able to see the blue dots, or whatever colour you wish to use, would be small dots, which would mean you'd be able to see the presence of the xylem vessels. Alternately, you could apply a stain like fluoroglutenol, which would stain the lignified tissue, which is the xylem vessels, and then you could observe them under a microscope. But the key point is describe means what would you actually do in this case, and the answer is that you would put them into a dye or food colouring, then cut a transverse section. Transverse just means across. Alternately, you could cut a thin transverse section, then apply fluoroglutenol or a stain that attaches itself to lignin, and then observe under the microscope with a low power. So water starwort is a hydrophyte belonging to the genus Callitrichi. Goodness knows how you pronounce that. It's an aquatic plant which has its stems fully submerged in water. Now cholla is a cactus and it can grow over 4 meters tall. So they've given you an example of a hydrophyte and a xerophyte. So hydrophytes are adapted for life in water, and xerophytes are adapted for conditions where water is a very scarce resource. Also, they've told you that the cactus grows to over 4 meters tall. Now, in the space provided below, give one way in which you would expect the walls of the xylem vessels. So they only want you to talk about the walls. It's not the distribution, it's not the amount of the xylem, it's about the thickness, it's about how lignified those two, the walls are in the xerophyte and the hydrophyte. And they're asking you to compare those xylem walls to a standard mesophytic plant, so a herbaceous dicotyledonous plant growing in a deciduous woodland similar to that which we get in the UK. So how would the walls of the xylem vessels be different? Well, the water starwort would contain less lignin than the deciduous plants. The cholla would contain more lignified walls than the deciduous plants. Alternately, you could have said that the water starwort would have thinner walls and the cholla would have thicker walls. It's only referring to the walls of the xylem, not the quantity and the distribution of the xylem vessels. Xylem and phloem are two vascular tissues found in plants. So state one similarity and two differences between the structure of xylem and phloem. So similarity, you need to say that they both are something, or they both have something. So something you might consider is that they both consist of cells which are stacked on top of themselves. Another similarity you might consider is that neither of them have a nucleus. There's a nucleus in the phloem companion cells, but not in the sieve tube elements. Also, you might consider that they are both tissues. That means they're made of a number of different cell types. So similarities, they're both made up of cells joined end to end. Xylem vessels and sieve tube elements both lack nuclei. You must refer to the sieve tube elements, not just to the phloem. And both are complex tissues because they're made up of more than one cell type. Now, differences must be a comparative statement. So you must say something like, xylem contains lignin, whereas phloem does not contain lignin. You can say xylem has a wide lumen, however, the phloem lumen is narrower. The lumen is the size of the hole in the tube. Xylem has no end walls because it has no sieve plates. However, the phloem has sieve plates which are end walls with small holes in them to allow the cytoplasm to spread through to the next phloem sieve tube element. Xylem does not have companion cells, whereas phloem does have companion cells. There are no sieve tube elements in the xylem. There are sieve tube elements in the phloem. There are pits in the sides of xylem vessels. There are no pits in the sides of phloem vessels. And xylem has no cytoplasm and no organelles, whereas phloem does have cytoplasm and a few organelles. Needs to be a comparative statement which is referring to the same feature or the absence of that feature in one tissue as opposed to the other tissue. Bony fish and insects have different gas exchange systems. So remember, insects have tracheae or tracheoles, and bony fish have gill lamellae, which is where they exchange the oxygen and the carbon dioxide, and that those gill lamellae are mounted on gill filaments, which form gills, which are the organs. So both can be observed by dissection, which is cutting up the fish or the insect. Now, describe how you would carry out the dissection to display maximum detail of either gas exchange system. Now this is a somewhat odd and a little bit confusing question. What they actually want you to say is, if I got hold of a fish, what would I do? And hopefully you would cut away the operculum. And the operculum is the hard flap which covers the gill. You'd cut that away and then you'd observe the gill. You'd cut out the gill perhaps and put it onto a binocular microscope, and perhaps you'd flood the gill with water so that the gill lamellae would come away from each other, so you'd be able to see maximum detail. Alternately, you'd cut away the exoskeleton of the insect and then perhaps put it under a binocular microscope, flood it with water and prod around with a mounted needle so you could see the presence of the tracheoles. So the mark scheme wanted you to say that you would remove the operculum or cut away the exoskeleton, and you specifically had to refer to these words, and then any method to observe or display the gills or the tracheoles, so putting it under a binocular microscope and flooding it with water and poking around would be acceptable in that case. So on to part B. So part B tells you all about an experiment that a student did where they took 3 different beetles, and they were from the same species, and they placed them in thermostatically controlled water baths, and they observed them for an hour, and the number of times the beetle surfaced to renew its air bubble was recorded, and then they calculated mean values for each temperature for each of the 3 beetles, and then recorded to the nearest whole number. Now that's an important part of the answer. And then they show you the results. And after you've read all of this detail about beetles and all of this detail about the practical, you're thinking, well, they're going to ask me about this practical investigation, and they don't. All they say is that the student made an error in their working. So they've got the temperature, which is 10 degrees, then they've got the 3 beetles, and then they calculate the mean. And they ask you to put a ring round the error in table 3 and then write the correct answer next to it and use the space below to show your working. You must show your working. It's not sufficient just to calculate it. So where's the mistake? Well, for those of you who are fairly observant, you'll see that the mistake is in this row here. And the way you can work it out is that if you work out the mean for 10 degrees, well, it's 10 plus 12 plus 8, which gives us 30, and we divide by 3 because there are 3 beetles, and that gives us a mean of 10, so we know that that's correct. And then if we take 18 and add 22 and add 18, we get 58. And if we divide by 3, we get 19.3. So the nearest whole number is 19, so we can say that this is incorrect, and we need to write 19 next to it, and then this is the working that we needed to show. And then 44 plus 48 plus 38 gives us 130, and we divide by 3, and that's going to give us 43.3. So that's again the correct answer of 43, so that we know that now the correct one is that the mean of 20 is incorrect and that 19 is the right answer, and that we should have put this in as our working. So figure 3 shows a diagram of part of the gas exchange system of an insect. So we've got trachea, and then we've got the muscle, and then between the trachea and the muscle we've got small ends here which are blind ending, like cul-de-sacs, where the trachea splits into tracheoles, and those tracheoles go into the muscle cells to supply the oxygen and to allow the carbon dioxide to diffuse out. Now along the trachea we've got these thickenings. These are made of chitin, and the role of that is to keep the trachea open and stop it collapsing. And on the outside we've got a spiracle, and that's a valve which can open and close to maintain the humidity of the inside and allow to help with the ventilation of the insect. So name the structure labelled A. Well, it is a tracheole. So describe how the trachea of a mammal, such as you, is different from the trachea shown in figure 3, which is of the insect trachea. Now, describe just means tell us what the difference is. You don't have to say anything about what it does or what it's made of. It's just how it's different. So all they're looking for here is comparative statements, such as mammals have just got one, whereas insects have lots of trachea because they have lots of spiracles down their body, so they have lots and lots of trachea. Mammals have a much larger diameter, insects have a much smaller diameter, so that's a comparative statement. Mammals have cartilage, whereas insects have chitin, or they don't have cartilage. Mammals have a C-shaped ring of cartilage because behind that you have the esophagus, so it enables you to swallow food, so it's an incomplete circle, whereas insects have a spiral support which runs continuously around the whole tube. Mammalian trachea are much longer, individual insect trachea are shorter. The trachea in mammals branch into bronchi. You've got 2 bronchi going to each of your 2 lungs, one into each lung, and insect trachea branch into tracheoles, not into bronchi. And then you might say that the mammalian trachea has things like smooth muscle to control its diameter, has goblet cells to secrete mucus, it has ciliated epithelium to clear the mucus, whereas individual insect trachea do not. Remember, the statements must be comparative. Alveoli are located in the lungs of mammals. Explain how alveoli are adapted for efficient gas exchange. Now, you've got to be really careful when answering this question that you only refer to alveoli. It's not about diaphragms, not about ventilation, it's only about alveoli. And then the explain bit is tell us the scientific principle that is important here. So you might say that they have lots of alveoli to give you a very large surface area for rapid diffusion, and that means that it is efficient gas exchange. Alternately, you might say that there is only a single layer of cells around the outside of the alveoli, giving you making them very thin, so that there's a very short distance for diffusion of oxygen and of carbon dioxide, hence it's efficient. That they are elasticated to allow them to expand, which gives you a larger surface area, which makes it more efficient. So all the time it's only referring to alveoli, it's the specific structures in the alveoli and why that makes efficient gas exchange, because it makes efficient diffusion. So you've got to be very careful when answering this question to get the 6 marks. So looking at the mark scheme, so for level 3 you need more than one correct adaptation and an explanation of how that adaptation improves efficient gas exchange. So the adaptation might be that there is a very large surface area because there is a large number of the alveoli. Why that's important? It gives you a high rate of diffusion. It's got a surfactant, so you secrete a glycoprotein which reduces the surface tension between the water molecules, which are meaning that you've got a moist surface to the alveoli, and that reduces the cohesion between the water molecules, and that prevents the alveoli from collapsing. That's the elastic fibers stretch and recoil. When they stretch, the whole of the alveoli increases in surface area so that you have more surface area for efficient gas exchange, and then when they recoil as you exhale, then that helps to force air out, which will ventilate it, which gives you more efficient gas exchange. Then other things you might want to think about would be that there are thin walls to the alveoli, because they are only 1 cell thick, that they're made of squamous epithelium, which is sort of flat. They're like floor tiles for cells, and those flattened cells mean that the capillaries are very close to the alveolar walls, therefore you have a short distance for diffusion, which gives you a high rate of diffusion, which makes it efficient. They then go on to talk about maintenance of the diffusion gradient, that the fact that the alveoli, because it's ventilated, it means you constantly replace the oxygen, constantly remove the carbon dioxide, that you've got a well vascularized tissue, that there are lots of blood vessels, that those blood vessels are close to the alveolar wall, therefore you're constantly replacing the blood, which is meaning that you have a constant diffusion gradient from the alveoli into the blood, and for the oxygen, and for the carbon dioxide to diffuse out of the blood and into the alveoli. So all the time you've got to be thinking, I'm specifically thinking about the alveoli, I'm only making points about the alveoli, and then I'm linking that to how that adaptation means that this leads to better gas exchange than if it wasn't there, which is explaining why that increases the efficiency of gas exchange. So lymphocytes form an important part of the specific immune system in humans. They can be classified into B lymphocytes and T lymphocytes. So for each of the statements in the table below, use ticks or crosses to identify whether the statement belongs to a B lymphocyte, a T lymphocyte, or to both B and T lymphocyte. The first one's been done for you, so you don't get any credit for that. And the rest, it's 1 mark per correct row. So do B and T lymphocytes form part of the immune response? Well, yes, they both do. Do they differentiate into memory cells? Well, yes, they do. You get B memory cells and T memory cells. Do they produce chemicals that can cause the lysis, which is the bursting of infected cells? Well, T lymphocyte do, but B lymphocyte don't. And which ones form a plasma cell clone? Well, B lymphocytes, remember, differentiate into plasma B, which make the antibodies and memory Bs, and T lymphocytes don't form plasma cell clones. They do differentiate into other ones, but they don't form plasma Bs. So that one is incorrect for T lymphocytes. So it was 1 mark per correct row, not per tick, per correct row. So figure 4 shows the concentration of antibodies in a patient's bloodstream following an influenza vaccination, and then a subsequent infection with the influenza virus. So we've got concentration of antibodies on this axis, and then we've got time along the bottom. So a vaccination is exposing your body to the antigen without exposing it to the pathogen. So when you carry out the vaccination, what's going to happen here? Well, we're going to have the primary immune response, which is clonal selection and then clonal expansion. So it's about finding the correct T cell and B cell which possesses a complementary receptor on the surface which is complementary to the antigen which will be being presented by the antigen presenting cells after the vaccination. Once you've found that correct B and T lymphocyte, well, they divide and differentiate and you produce plasma B cells, which leads to the concentration of the antibodies going up. But you do have this delay where clonal selection takes place, and then you've got clonal expansion, the production of the plasma cells, which leads to an increase in the number of antibodies against flu, against the flu antigen in your blood. Now, when you subsequently, and this could be a few days, a few weeks, or a few months later, when you get infected by the flu virus, you're getting infected by the pathogen. And the pathogen has on its surface the same antigens that you used in your vaccination. So that the second time you experience the antigen, you already have the memory cells, so that the concentration of antibodies rises far more rapidly as a result of the second time you're experiencing the antigen. So the first time you experience it on the vaccine, you're going through clonal selection, clonal expansion, producing the memory cells. And the second time you experience it, you've already got the memory cells, so that all they need to do is to divide to produce more plasma B cells, which can then produce much more antibodies far more rapidly, which is why this gradient is much steeper here than it is here, and also it reaches a far higher concentration the second time you experience the antigen as opposed to the first time you experience the antigen. Now, this is a classic graph that you must be able to explain. It usually comes up at least once in an AS paper, and the question asks you to describe where the primary and secondary immune responses are taking place on figure 4, and explain the differences between the two processes. So describe where they're occurring. Well, the primary immune response is occurring here, and the secondary immune response is occurring here. And then explaining the differences. Well, the major difference is that primary immune response has to undergo clonal selection and clonal expansion because it's the first time you've experienced the antigen. Then the secondary immune response requires the expansion of the memory cells, so the division of the memory cells, and then producing plasma cells from those memory cells that you've already produced as a result of the primary response. So looking at the mark scheme, firstly, where do they occur? So the primary response is just after vaccination. The secondary is just after infection. And explaining the differences in the response is that the primary response is slower because of clonal selection and clonal expansion. Therefore, you produce fewer antibodies more slowly, and the secondary response is quicker because of the presence of the memory cells, and that produces more antibodies more rapidly. So this is a 6-mark question about the spread of Ebola in West Africa. Now, you're unlikely to get a question that's identical to this, so the major learning point is how to approach these questions and how to think about the factors and then suggest what actions could have been put in place. So looking at the question to start with, it says a number of common factors affect the spread of communicable diseases in humans, and some of them are relevant to the spread of Ebola. From the information above, so looking at the bullet points above, discuss these factors and then suggest what actions could have been put in place which would have addressed those actions which were causing the spread. So looking at the information above, what was increasing the spread of the Ebola virus, and then what would you suggest that could have been done to counteract those actions which are making it spread more easily? So it says in West Africa during 2015 there was a serious outbreak of the viral disease Ebola. You don't need to know anything about Ebola, but they do tell you it's spread by contact with infected bodily fluids. That's really important. And then looking at the individual bullet points. At the start of the outbreak there was a severe lack of trained health workers. So perhaps an action to improve that would be to train more health workers. Much of the nursing was carried out in the family home. Well, if we link that to the contact with infected bodily fluids, then it's more likely to spread it to family members. So maybe a suggestion would be to treat them away from the family home. Many residents lived in close proximity to one another, and sanitation was often of a poor standard. So why would that cause an increase in transmission? Well, it's coming into contact with infected bodily fluids. How could you improve that? Put in better forms of sanitation and disposal of bodily fluids. In times of illness, it was common for people to travel to stay with close relatives, often in nearby villages or towns, and then as the outbreak spread, some residents left their villages to flee from the disease. So that's spreading it to new unaffected areas. That's meaning that you're going to have outbreaks in new villages. So you could address this by restricting travel, introducing a travel ban. Local mourning and burial practices involved gathering at the family house to pay respects to the deceased. The deceased is the one that's died of the disease, which is caused by infected bodily fluids, and then prior to the burial, the deceased was usually bathed by close family members, which again is a remarkably bad idea. So this needs to be addressed in terms of trying to alter the culture that's associated with dealing with the deceased. Now, the learning point from this is to connect the contact with infected bodily fluids, the concept of communicable diseases, and then to suggest specific measures which could have been taken which addressed the problems that caused the disease to spread more rapidly. So to get level 3, that is to put you into the 5 to 6 mark band, you need to discuss more than one correct factor related to the information in the rubric of the question that is causing an increase in the transmission of Ebola virus, and then plausibly suggest an action that could be taken to address that factor and reduce the transmission. This point about ignore repetition of bullet points and suggestions that are merely reverse actions is very important. So you can't just repeat the exam rubric back to the examiner. You have to say why that is a problem and then how you would address it. So for an example, the lack of trained professionals, therefore there is a lack of treatment, or there's a lack of understanding in the way in which it's spread. And the possible action would be to send more trained health professionals into the area, better access to hospitals, train more professionals, educate the population so that they can take the necessary precautions. And then the caring for the ill by family members. Why is that a problem? Because they're exposed to the pathogen, because they don't have safe nursing techniques such as hand washing or surgical gloves, and you can spread the pathogen more easily. Therefore, instead of caring by family members, you should care with trained health professionals who know how to reduce the spread of the pathogen. Overcrowding and close proximity. Why is that a problem? Because they could be spread through droplet infection and could spread more easily within the community. The action would be to have less sharing of rooms, or to use larger rooms, or to improve the ventilation. Now, all of these are principles that apply to all communicable diseases. They're just using Ebola as an example of a communicable disease where these specific problems are occurring. So the poor disposal of wastes, why is that a problem? Because you can pick up the pathogen from the feces. Possible actions, education projects, sewage disposal, use of hand washing gels, Next factor, that they can flee from places with the disease. Why is that a problem? So the pathogen can spread to a wider area and the epidemic will become a pandemic. Well, hopefully it won't become a pandemic, but there is a chance that it could be, and that you can't then vaccinate the people or treat the people who fled the area and gone to a new area. So the possible action would be a travel ban or restrictions or health checks at airports or bus terminals, and then the quarantining of people or the use of police to prevent people from travelling. And then finally, the burial practices. Now, mourning and burial practices are difficult to change. The problem is that it brings people into close contact with the pathogen as it's spread by touch and bodily fluids. So there are possible alternatives, cremating the people who've died of Ebola, and then involving local leaders in promoting a change to a safer practice and an understanding why this long-seated practice is a bad idea when it comes to controlling the spread of a communicable disease. Now, having a look at what you needed to get, so for 5 to 6 marks you needed to expand on more than one bullet point, and you needed to get more than one correct factor that would affect the spread of communicable diseases in humans, and then apply a plausible suggestion of the action that you could do to address one of those factors. It needed to be a well-developed line of reasoning which was clear and logically structured, and it needed to be relevant and substantiated. Remember, with these questions, they tend not to be asking specifically about the disease, so it's not really asking about Ebola, it's more asking about the concepts of communicable diseases and getting you to apply those principles about preventing the spread of the pathogen and preventing the infection of new people, and applying that specifically to the example of Ebola. A study was carried out on butterflies in 2 different habitats in the north of England. The 2 habitats were farmland and mixed deciduous woodland. Now, the surveys were completed in 1992 and then at 4-year intervals, and the 4-year intervals are 1992, 2000, 1996, 2000, 2004, 2008 and 2012. Data were collected from butterfly transect sites in both habitats. Using this data, the total butterfly population in each habitat was estimated. Now, when you read the exam rubric like this for an exam question, it's very important to think, what have they not told me as well? What's missing? What detail is missing here that is going to be important later? And one of the most important things here is that they've told you that the data was collected from butterfly transect sites, but they haven't told you how many sites, how frequently it was done at each site at each 4-year intervals, so how many repeats there were. They haven't told you the method of sampling. They haven't told you that the sampling was done at the same time of year or in the same places. So this point needs to leap out at you as an immediate point that you're thinking, well, they haven't told me sufficient detail about this survey for me to reach a good conclusion. Now, in 2012, the general population of the butterflies in these 2 habitats reached historic lows as a result of the prolonged cold and wet weather. So this is only in 2012 you're reaching historic lows because of the weather. However, looking at the data, you can see that the decline starts in 1992 and reaches a historic low here. There is only cold and wet weather in 2012. So you're thinking, well, perhaps the decline from 1992 to 2012 is not caused by the cold and wet weather, it's caused by other factors. Then they go on to say between 1992 and 2012, the woodland had become overgrown due to lack of active management. In particular, the large number of spaces in the woodland had decreased. So the number of open spaces, in the woodland had decreased. Now, what's the consequence of that is you're getting a change in the habitat diversity. You're getting a different number of niches. Understory plants are becoming overgrown. There's lack of open spaces. So perhaps you're having fewer different niches within the woodland, which may lead to the decline of species that you see in the woodland here. Then when you come to look at the data, it's good to mentally read through what's going on. So you might say, well, in 1992 I've got 110 butterflies per square kilometre, and that falls in the farmland to around 80, whereas in the woodland it's about 98, and that falls to about 48. So I've got a much larger percentage fall in the woodland than I have in the farmland. And having read through that in your mind, having read the rubric, now's the time to then go on and have a look at the questions that they then ask you about the data and about the survey. Calculate the total percentage decrease in the number of butterflies on farmland between 1992 and 2012, and then show your working. So we need to look at the number of butterflies in 1992, which is 110. We can read that off the graph. And then how many there were in 2012, which is 79. Now you do have to be very careful when reading these off the graph. It's between the two lines. Each line is 2 butterflies per square kilometre, so this gives us 79. Now the percentage decrease is the number it started with minus the number it ended up with, which is 110 minus 79, and that gives us 31, divided by the original multiplied by 100, and that gives us 28.18. Now they do ask you to give your answer to the nearest whole number, and the nearest whole number here is 28, so the answer is 28%. Now this question's got a crucial phrase that must leap out at you and that you need to learn from. And the crucial phrase is using the data. Now, using the data means there will be credit given in the mark scheme for quoting data or doing calculations based on the data that they've given you. And then compare the changes in the number of butterflies on farmland and woodland between 1992 and 2012. So looking at this, you might think, well, the most obvious change is that the number of butterflies on the farmland falls far less than the number of butterflies on the woodland. And the number of butterflies on the woodland falls very steeply between 1992 and 2004, and then falls less steeply, and in fact about the same rate between 2008 and 2012 in both of them. So the first 3 correct points in the mark scheme is that the number in the farmland stays higher than the number in the woodland, so it's a comparative statement about the numbers of butterflies. The number of butterflies in the woodland has a greater decrease than those in the farmland. Then from 2004 to 2012, they both fall by about the same amount. Now, at the start of the question, you did a calculation which showed you that there was a 28% decrease in the farmland population. Because there's a using the data point in this question, if you did the same calculation on the woodland population, you'd see that it went from 98 down to 48, which was a percentage decrease of 51%. And they credited you if you did that calculation and then compared that to the 28% and said that that was a difference of 31 per square kilometre. And it's important then to use that data. So both habitats experienced the same weather conditions. Now they ask you to suggest a reason for the difference in the rates of decline in butterfly numbers in woodland and farmland. Now suggest means come up with any reason that is reasonable. But a reason you can't suggest is the severe weather, because they both experienced the same weather conditions. So the differences must be something to do with the nature of the habitat. So you could have suggested that the woodland population dropped by more than the farmland population because there were more predators or parasites or diseases, or more interspecific competition, or that because of the lack of management there was a loss of habitat or a loss of food supply, a loss of breeding sites. It's not just enough to say there's a lack of management and a lack of open spaces. You need to say the consequence of that, which is the loss of habitat, food supply or breeding sites. And then the farmland could have decreased less because there might be wildlife refuges. They might have conserved hedgerows. There might be fewer predators or fewer parasites or more open spaces for breeding. Now suggest means come up with any reasonable answer that could have explained this, but put it in sensible A-level biology terms. So a student made the following statement: these data show that the changes in butterfly numbers was caused by changes in the weather conditions in England. Now it says England, but remember this study was only done in the north of England. So comment on the validity of this statement. So this is where reading the rubric of the question was extremely important, because this statement isn't valid because the weather conditions only apply to 2012, and the numbers were falling before 2012. And there may not be a link between the weather conditions and the butterfly decline. They may be an association, but it may not be causal. You don't have enough data, you don't have enough evidence to know that it's the cause of the decline. You know that there is poor weather and there is a decline in butterfly numbers, but you certainly don't know that this is a causal relationship. And finally, that the weather conditions in the north of England don't represent the weather conditions in the whole country. So state one variable that scientists should control when carrying out surveys such as this. So put yourself in the position of a scientist who was carrying out a survey of butterfly numbers. Hopefully you'd think, well, I'll do it at about the same time of year each year. I'll do it at the same time of day, because the seasons and also the time of day are going to affect the number of butterflies that are in the area. So the variables they suggest that you should control is that you should do the survey at the same time of year, same time of day, have the same size of the area of sampling, same length of transects, same number of transects, use the same sampling technique, or do it in exactly the same place in each habitat. All of these are sensible variables that you would control if you were carrying out the survey yourself. Butterfly species in severe decline on farmland include gatekeeper, large skipper. I won't list them all, but there are 7 in total in severe decline. Butterfly species in severe decline on woodland include brown argus, common blue, and there are 11 in total. Now state which habitat you'd expect to have the greater species richness, which is the species richness is the number of different species in the habitat, and give 2 reasons for your answer. Now this needs really careful reading, because it says greater species richness, but it's not greater species richness of butterflies, it's greater species richness, full stop. So you do need to think about that in your answer. So you could have made a case for woodlands having a greater species richness or farmlands having a greater species richness. You just have to justify your reason. So there was no right answer on which one would have a greater species richness. It was just the reasons that were the important bit and got credit. So woodlands could have a greater species richness because they've got a greater number of butterfly species in decline. So they've got 11 in decline as opposed to 7. So there were probably a greater number of species present. Additionally, there are just more species there, so there's a greater species richness. Well, why might that be? It's because there are more niches within a woodland than there are in a farmland. Or there might be less use of pesticides. Or farmland is likely to be a monoculture with a limited number of plant species. You're only growing one crop like wheat or barley or oats. And therefore, you've got fewer plant species, therefore there are fewer niches and a lower variety of food plants, so you have less butterflies. Alternately, you could have said that farmland has a greater species richness because it's lost fewer butterfly species. It's lost 7 as opposed to 11. Therefore, you might have a larger number of species still there. You could have conservation areas within the farmland, such as hedgerows or wildlife refuges, so that there would be more niches in these peripheral areas where there were more food plants, therefore more niches for more butterflies. Finally, remember that this question says state which habitat you would expect to have a greater species richness, but it doesn't say of butterflies. So the general point here is that butterflies are an indicator species of what is happening, so that then because there are more butterflies, it may show that there are more other species within that habitat. So there are female silver-washed fritillary butterflies, and they're usually orange-brown colour. However, a deep olive-green colour can be seen in some females, largely in the south of England. So what's the term given to this type of biodiversity? Well, this is a genetic biodiversity because this is caused by genetic differences in the butterfly species, so genetic biodiversity. Now, give one possible benefit to the species of this type of biodiversity. So why might this be important to the species? What advantage could it give? Well, it gives an opportunity for adaptation. It allows adaptation to the environment. If the background colour of the environment changes, then it will give you camouflage to predators, and it gives you genetic variation on which natural selection can take place. So if you had in the south of England more olive brown background plants, then the olive green background plants, then the olive green butterflies would be at a selective advantage over the orange brown butterflies because they would be more cryptic, therefore there'd be less predation. So you need to think about, with these answers, what's the benefit to the butterfly? And it's that it allows adaptation, it provides variation for natural selection, and then offers camouflage, an opportunity not to be predated. Therefore there's an advantage to the butterflies of having that biodiversity. A group of students decided to investigate the glucose content of 3 types of fruit juice. They carried out the Benedict's test on known concentrations of glucose solutions and used these to calibrate a colorimeter. Now this is known as producing a calibration curve, and you're then going to use that to test the absorbance in the fruit juices and then be able to read off that curve the concentration of the glucose solution. So they put a glucose concentration of 1 millimole per decimeter cubed, they boiled it with Benedict's, and then they shone a light through through a filter, and then they looked at the percentage absorbance, and they saw that you had the highest percentage absorbance with the lowest concentration of glucose, whereas when you've got the highest concentration of glucose, you've got the lowest percentage of absorbance. And this is because the more glucose you have, the more it's reducing the copper 2 to copper 1, so the less blue it's going to be, and the more the precipitate is going to settle. So it's going to be the bluest when you have the least glucose, and that's going to give you the most absorbance. Now they ask you to plot a graph of the mean percentage absorbance at each glucose concentration. Now when you're plotting graphs, you've got to take a lot of care to get the axes right. So the labels for the axes need to come off the table, and they need to be the column headings. So the independent variable, which is the glucose concentration, this needs to go on the bottom, and you need to put glucose concentration in millimoles per decimeter cubed. You've got to put the unit, and then because you're plotting mean percentage absorbance, you need to put that as the dependent variable on the up axis, so it needs to be mean percentage absorbance. So for the first mark, you had to have an appropriate scale. Now, appropriate scale is a scale which would mean that the graph occupies more than 50% of the graph paper, because if it was less, then you could have made it twice as large. There's no need to distort the intervals to give you some hard to plot intervals. It's perfectly acceptable to have it occupying 2/3 of the graph paper. Now this is the graph, and which is the actual point that you've plotted. Now you've also got to label the axes correctly, so you put the independent variable on the bottom and the dependent variable on the up axis, and you take the labels from the column headings in your table. So you've got to put mean percentage absorbance, not percentage absorbance, and you've got to put the unit of the independent variable. Then you get a mark for plotting the points correctly, so you need to be within half a square, and they do check, so you've got to spend some time making sure that you plot them correctly. And then you should draw a straight line of best fit. Now I'm not quite sure what they mean by that, but hopefully what they mean is something that looks like this. But most crucially, it needs to not extend beyond where you have points. So for some students, the temptation is to do this. Please don't do that. So just only when you draw a graph, only draw a line through where you have data. You could probably also have just joined the dots with straight lines, and that would have been perfectly acceptable, but it wouldn't be acceptable to do a wibbly curve through it. So do a straight line of best fit, don't go beyond where you have points. So you made the calibration curve through using known concentrations of glucose and putting those in the calorimeter after you've done the Benedict's test on the known concentrations. So now what do you do with it? You've got to explain how the students would use that calibration curve to estimate the glucose concentration of the fruit juices. Now the students have got 3 different fruit juices. They carry out the Benedict's test on each fruit juice, and then they put them into the calorimeter. So the calorimeter is going to give you an absorbance value. So if the calorimeter for fruit juice A gave you an absorbance value of, say, 52%, then you would go from 52% and you'd go out until it hit the line of best fit, and then you would go down to see where that reached on the axis of glucose concentration, and then you would read it off from that axis. So in the mark scheme, they wanted you to find the absorbance of the juice using the calorimeter, and then find the concentration that corresponded to that absorbance. And the way you would have done that would be to look at the absorbance value on the y-axis, then go out to the line and down, and then read it off the x-axis to give you the concentration of glucose in that fruit juice. So the students wrote the following hypothesis: the higher the concentration of glucose in the fruit juice, the sweeter it will be. Now describe how you would carry out a controlled experiment to test this hypothesis without using a colorimeter. So for this hypothesis, you need to know the concentration of glucose in the fruit juice, and then you need to make a judgment about how sweet it is. Now it says it's a controlled experiment, so there'll be a mark for suggesting a controlled variable. Now you have to test the glucose concentration without using a colorimeter. So how could you do this? You could use a biosensor, such as you get in blood testing diabetes kits, or you could use a Benedict's test, but make a color chart of the different range of Benedict's test results, which might range from green to a bright red color. So the first thing you have to do is to make a judgment of sweetness. So you're going to have to taste the fruit juices. And then you need to take a sample of each and put it into a biosensor and take the reading, or test it with a CliniStick, which will give you a range of colours, or test it with the Benedict's and observe the colours. Now crucially, you do need to say and take the reading and observe the colours. And then you might get a rank order for sweetness and for fruit juice for glucose concentration, and then you might compare the rank orders for if A was the sweetest, did A also have the highest glucose concentration? And then you'd get a mark for saying how you would control any single variable. I mean, maybe temperature, or the tester, or the time of day that the test was done, or what the tester had eaten beforehand. Just anything to say how you would control it. So suggest one reason why the results for this experiment might not support the student's hypothesis. And remember, the hypothesis is that the higher the concentration of glucose, and it's just glucose, then the sweeter it is. The big problem with this is that sweetness is a subjective judgment. So one person might think that fruit juice A was the sweetest, another person might think that it's fruit juice B. So it's very subjective, it depends on taste buds. Additionally, this hypothesis refers to glucose concentration, but there are other sugars which affect the sweetness, and those other sugars are present in those fruit juices. So you're not just looking at the glucose concentration, there are other sugars present. So you could have said that tasting is subjective, it only gives you a qualitative judgment, not a quantitative. It's hard to quantify sweetness, different people have different judgments, opinions or taste buds. That the colour judgment when you do the Benedict's test is again subjective, so this is when you're calculating the glucose concentration without using the colorimeter. And then additionally, the fruit juice may contain sucrose or fructose, or possibly an artificial sweetener which may affect the sweetness, but the glucose concentration may be lower. So glucose and cholesterol are both molecules transported in the bloodstream that may need monitoring in people with different medical conditions. And we've got figure 6 representing the structure of a cholesterol molecule, and here is our cholesterol molecule. Now state 2 ways in which the molecular structure of cholesterol is similar to the molecular structure of glucose. Be careful with these state 2 ways questions, they only mark the first 2 answers, and it's only looking for the molecular structure. So all they're asking for you to say here is that they both contain carbon and hydrogen, and they both contain oxygen, and additionally they both contain a hydroxyl group here. So the ways in which they're similar: both contain carbon and hydrogen, both contain oxygen, and both contain a hydroxyl group. That's it. And here's the mark scheme. So finally, Glucose is an important biological molecule required by cells for cellular respiration. State the physical property of glucose that allows it to be easily transported in the bloodstream. Well, the bloodstream is mainly water. Plasma is mainly water. So what's the physical property that allows it to be easily transported? Well, it's soluble in water. And that was all they wanted, was that it was soluble in water, or that it dissolved in water, or that it was polar so that it dissolved in water. So that's the end of the paper. I hope you found it helpful. I hope you've enjoyed this guide to the paper, and good luck with your exams.